Good morning, everyone. So hopefully today no power outages because we have quite a bit of content to get through. I had a nightmare last night that all of you walked out of this lecture because you hated it and you were really mad at me because it was too hard. If you thought things were difficult up until this point, let me tell you, shit's going to get real today. You can get through your degree as a student without knowing what the cerebellum is, but you cannot get through your degree without knowing what an action potential is. And that's what we're going to learn today and on Friday. So we are going to spend 2 whole lectures on the electrophysiological properties of neurons. Trust me, if at the end of Friday you don't know what's going on, you really need to make sure you know what's going on. And what that means is you need to make an appointment with your TA or myself and talk yourself through it because it is challenging, especially for those of you that lack the biological background. It's going to be hard. A lot of you are not going to understand it right off the bat, and that's totally and completely normal. So I'm going to take this sucker slow. I'm going to repeat myself many, many times. Some of you are going to be bored, maybe 3 of you out of 110. For those 3 of you, I'm sorry. Feel free to leave. But I'm doing this for the rest of you who I'm sure are going to struggle with this content. Okay, so make sure you've got some caffeine on board, or meth, whatever is going to get you through here, because it's going to be challenging, okay? Please don't walk out. Sad prof. So you go from having exam anxiety dreams, I'm sure you've all had them, you show up to the exam, you're naked, or it's a stats exam, you studied for 2200, whatever. I have prof anxiety dreams. I show up to my exams, there's a tornado, and I can't administer scantrons. It sucks. So I'm going to try to give you kind of an overview before we get into the nitty-gritty of action potentials. What you should know by this stage of the game is that our nervous systems are geared to do sensory motor activity. What that means is we take in information via our senses. So we take in visual information from our eyes. We take in auditory information from our ears, tactile information from our skin and our organs, taste, smell. This information comes into our nervous system via those afferent nerves and makes connections with all sorts of funky regions in the brain. And what we as humans and we as a lot of organisms on the planet, what we do is we make decisions on how to deal with that incoming sensory information. So we are a sensory motor, sensory in, motor out organism. Many organisms are, not most. So information comes in via our senses. So let's imagine that, for example, you have a bowl of ice cream in front of you. You have a number of different sensory cues, right? You have the sight of the ice cream. You might have the smell of the ice cream. You could probably touch the ice cream if you wanted to. What this means is that your ice cream neurons, the neurons that might code for the sight of ice cream and the smell of ice cream, the touch or the taste of the ice cream, they're silent up until that point. So those neurons are quiet. They're not transmitting any information. But suddenly we get sensory information that comes in, and those neurons, suddenly, they get excited. They want to convey a message, right? Because if we see the ice cream when we're hungry, what that might do is then we have the sensation coming in, and we have the perception of what that means to us, right? So we have, maybe if we're hungry, the perception is, oh my God, I really need to eat that ice cream. If we just ate 3 Big Macs, the perception might be, oh God, I can't possibly eat dessert. But sometimes we overwrite that anyway, right? Because we're like, yum, ice cream. If we're on a diet, maybe we'll say, no, I just can't, I need to eat my salad, right? So each of us may react differently, but the point is, we all have those ice cream neurons that are firing in response to seeing the ice cream. So we have some magical thing called perception, which you'll learn in 3202, not in this class, and this leads us to action. Do we approach the ice cream and eat it? Do we back away from the ice cream and say, no, I'm being good, I'm on a diet, or no, I can't possibly eat it, I'm totally full? But you have neurons that are that are being activated at each of these steps. Neurons that are normally silent suddenly spring to life. They spring to life at the part of sensation. They spring to life at the point of perception. And they also spring to life when we have to make actions to direct us, approach, or avoid something. So we have motor neurons that are then being active. So hopefully this is going to give you a bit of an overview to what it means to have neurons that are active, right? We hear that term bandied around. I'm sure I've said it all throughout the term so far. What does it mean to have a neuron that is firing? What does it mean to have a neuron that is active? And so that's what you're going to learn in today's lecture and next class. So before we get into the nitty-gritty of neurophysiology, we need to be reminded of the basic structure of a neuron, which is outlined here. This is from a different textbook. But essentially, the take-home message is that, as you've learned, we have neurons that communicate with one another. And each neuron is what's called a presynaptic neuron, a sending neuron. It sends a signal. And it's also a receiving neuron, a postsynaptic neuron. So in this case here, the neuron that's in blue is the presynaptic neuron. It's the one that is sending a signal. And you can see the axon terminals are making contact with that postsynaptic neuron in yellow. Okay? So the question is, what's happening? What is occurring in this magical, magical space that is the synapse? What exactly is being sent? What message is being sent by the presynaptic neuron to the postsynaptic neuron? So we're going to learn about the electrical properties of the neurons today. Next week, we'll learn about the chemical properties. A little bit more biological detail about the cell itself. Every cell in the body, be it a heart cell, liver cell, skin cell, neurons, has what's called a cell membrane. The membrane it acts like skin. It is the separation between the outside of the neuron and the inside of the neuron. It is known as the phospholipid bilayer because the cell membrane is actually consisting of these phospholipid molecules here. You can see each of these round circles represents the head of the phospholipid molecule, and each of these little vertical things here are the tails of the phospholipid molecule. The phospholipid head is hydrophilic, meaning it likes water. Philis means love, hydro means water. The tail regions here are hydrophobic. It repels water. So what that means is the phospholipid bilayer, two layers, naturally forms because these tails repel water, so they go from being like this to like this, right? So the tails are kind of attracted to each other here. They come into the phospholipid bilayer because they're repelling water. And that's because the intracellular fluid, the fluid that is within the cell, as well as the extracellular fluid, the fluid that is outside of the cell, is mainly based in water. So they're repelling these fluids and moving towards themselves. Okay? And because the hydrophilic heads are attracted to water, they're okay. They like to be making contact with water. So this is essentially the cell membrane. It's a phospholipid bilayer. It's formed by the properties, the chemical properties of these phospholipid molecules here. The important feature of the cell membrane is it regulates movement of molecules, ions, inside and out of the cell. It's protecting the inside of the cell. It wouldn't make sense like your skin, right? You don't want to be absorbing everything that's out there in the environment. We need to protect our internal environment somewhat. And that's what that cell membrane does. Okay, so it regulates the movement of substances inside and out of the cell. Most substances cannot pass into the cell. That said, there are mechanisms to allow things inside the cell or to allow things from inside the cell to outside the cell. And these are called channels. So we have openings in the cell membrane that allow the passage of what are called ions. I'll talk to you a little bit later about what ions are, but they're a specific substance that is highly important in the cell environment. So you can imagine the channels are literally like gates. Here is this channel, example of a channel. They're proteins, so they're made from those basic building blocks that make up our entire body. And they're embedded in the cell membrane, and they allow substances to pass only when they're open. Okay? So a gated channel, it's either open or closed. So here you can see sodium, for those of you that are savvy, Na+. Sodium is allowed inside and outside of the cell because the gate is open, and if the gate is closed, sodium is not allowed to pass in. If a channel is just always held open, it's called a channel, but they can be gated. Okay? Another type of protein that's found on the cell membrane is something known as a pump, and you'll learn about a specific kind of pump that's illustrated here, the sodium-potassium pump, shortly. And in this case, it's involving active transport, so we require energy. So a pump is different than a gate. A gate is if it's open, things flow inside and out of the cell readily. A pump, it requires energy. It physically gets molecules or ions inside the cell and pushes them outside of the cell, or it grabs ions from outside of the cell and pushes them inside the cell. Okay? So 3 types of proteins that are found on the cell membrane: channels, gates, and pumps. Okay? So be able to differentiate between these 3. Any questions so far? In order to try to understand the physiological properties, the electrical properties of cells in the brain, it is very hard to do this, especially early on in the studies that were completed in the 1950s, when we didn't have the kinds of sophisticated equipment that we have today. In addition, the neurons that are in our brains are very, very, very small. So early studies that looked to try to understand how these electrical signals were conducted used this organism, the giant axon. And the axon of these animals is much larger than human axons. Humans are 1 to 20 micrometers, which is 1/100th of a millimeter. And the squid is up to 1 millimeter. So, 1,000 micrometers. So it is quite a bit larger. And so as a result, we can use equipment, or the people that did these studies that back in the 50s, use equipment during that time that could investigate how axons conducted electricity. Anybody remember who was the first person I mentioned on the lecture of the history of the brain who determined that the neuron had electrical properties? Who was the first person? Someday you're going to have an exam, and you're going to have to know this. Starts with a G. Nope, Franz Gall did the phrenology. Yeah? Galvani, that's right. So Francis Galvani studied using the frog's legs. Remember, his assistant accidentally drew a spark on a scalpel and touched it to the frog nerve, and it jumped. So that's how we knew that the neurons had electrical properties. We just didn't understand how they were using electricity to send information. So these first investigations were done by Hodgkin and Huxley in the 1930s and the 1940s. So almost 100 years ago, which you can imagine how much things have changed since then. But they studied the giant axon of the squid to try to understand how it conducted electricity. So a few other pieces of equipment that we need to know about. The first is something known as an oscilloscope, which is illustrated here in this figure from your textbook. And an oscilloscope is essentially a voltmeter. What that means is it's measuring changes in voltage. So voltage is a measure of electricity, right? When you plug something into an electrical outlet in your wall, what's happening is you're having a transfer of voltage. And that voltage is going to power your hairdryer or your cell phone or what have you. So these oscilloscopes, what we can do is if we connect them to axons, and I'll show you how that's done shortly, is what it can tell you is what is the charge of that axon, right? What is the actual electrical charge of that neuron? And we can measure it in voltage. So for example, here in this cartoon, it shows you voltage in millivolts from around -80 to +30. And here we can see that the voltage is around 30 millivolts after we apply what's called a stimulus, which is S. So if we stimulate the neuron, we can see that it goes to about +30 millivolts. And here's an example of an actual reading, an output of a neuron. And you can see that it does fluctuate from very positive to very negative. Another piece of equipment that is important in understanding how we measure electrical potentials in cells is something known as a microelectrode. It's literally an electrode that is very small, hence micro. So it is used to record electrical activity from living cells. So you can see in this figure here that's from your textbook, the electrode, the tip, is placed directly inside the axon. So you can see here is a cartoon of this microelectrode coming into the cell. So importantly, the tip is in the intracellular fluid, and the rest of the electrode is in the intracellular fluid. So we're measuring the voltage inside the cell, okay? So this can be used to measure the neuron's electrical activity, or we can get output, or we can deliver a current, a charge, and we can therefore change the electrical potential of the cell. Okay, so we can record, or we can deliver an electrical charge. And this is known as stimulation. So when you hear me say, we stimulated this neuron, that's what we're doing. We're artificially making that neuron change its voltage. Yeah?
Is this what was used in the Montreal procedure, the ice melt burnt toast?
Yes, that's right. Yeah. So in the Montreal procedure, what Wilder Penfield was doing is he was taking something not unlike this and stimulating patches of the cortex. And what he was trying to get is, if I stimulate, artificially change the voltage of a neuron, can I find that neuron that leads to the olfactory sensation of burnt toast? So you're artificially yielding that same activity. So using our oscilloscope, our microelectrode, and our giant squid, we can get a sense of what is the resting membrane potential. What is the voltage of those neurons at rest when we have no presynaptic input into that neuron? Okay, because we've taken that axon and we've put it in a petri dish. Right, so it's not in situ, in the actual living organism. So we remove all inputs to that neuron, and we can ask it, okay, like that ice cream cell in our brain that is kept silent when we don't have ice cream. What is it telling us at rest? Okay, so here is that experimental setup. So here's our nice petri dish, and here's our neuron. And you can see that we have the reference electrode that is kept outside of the neuron, right? So it's in an artificial extracellular fluid. And then we have our recording electrode, and the tip is inside the neuron. And we connect these via an amplifier to our oscilloscope, which gives us an output of how that voltage changes when we put the microelectrode into the cell. And what you'll see is here, it's at 0 millivolts. We put the microelectrode into the cell, and boom, what do we get? Around 60, 65, 70 millivolts, minus 60, 65 millivolts. So what we're reading there, what that number is called the resting membrane potential. It is the electrical charge across the cell membrane. It's the difference in electrical potential inside versus outside of the cell. The outside of the cell has no electrical potential. It's going to read 0, more or less. As soon as we put that electrode inside the cell, it's going to read out on that oscilloscope or that voltmeter as minus 70 millivolts. So remember, this is just a reminder that the intracellular fluid is also known as the cytoplasm. You may have heard that way back in grade 11 biology. The extracellular fluid, the stuff that's outside the cells, is also known as interstitial fluid, which literally means in between. So it's the fluid that's in between the neurons or outside of the neurons. So we can conclude that minus 70, around that, okay, it's not always exactly minus 70 millivolts, is the resting membrane potential. Minus 70 millivolts is the resting membrane potential. Yes?
So the stimulus is what activates the channels which causes the sodium.
There is no
and
stimulus. There is no stimulus.
So what activates the channels then?
We're not even there yet.
Okay.
This is the resting membrane potential. This is absolutely baseline.
Okay.
There's no stimuli. This is just we take that neuron out of your brain and we pop it in a petri dish. Okay? Our ice cream cells completely quiet. Any other questions? Do you understand this? Do not be afraid to say no. If you don't understand this, the next step is going to be even more challenging. I'm going to give you 1 minute to think about it. Talk amongst your friends if you want. Tell them what you think the resting membrane potential is. This must be basic for you.
It's good though.
Done? Yeah, it's good. Good review. Yeah, Rachel. Why? I have no idea. I can't even. You guys dictate my pace. I don't. If you're in pain, go. Okay? Unfortunately, this lecture is make or break.
I understand all of it. There's just like a couple slides that I haven't learned before because I took a physiology class.
Okay. All right, any questions that's come out of it? Jen? No. That's Jen. Both blonde. Yes.
So I know
Let's not, don't even go there. Don't even bring that up. You will confuse everybody. Ask me a question about the resting membrane potential.
okay, is it the same between PNS and ENS? Because I know they're both related with different types of cells.
So she's asking, is the resting membrane potential universal across all neurons in the central and peripheral nervous system? Yes. Myelination has nothing to do with it. By the way, that reminds me, I told you guys astrocytes myelinate last class. A little bit wrong. Astrocytes don't myelinate. They aid with myelination. Okay, so it's the Schwann cells and the oligodendrocytes. Please take note of this, cameraman. Yes?
Is the electrical charge outside the cell always zero?
Is the electrical potential outside the cell always zero? Can I answer no for the purposes of this class? Just to understand the basics, say no. But it is not 100% true. Okay? Yes?
Sorry, what's that abbreviation MV?
Millivolts. Millivolts. Yes, millivolts. Anything else? Yeah.
The membrane, so it's charged, but is it like it's a negative charge?
Yeah, so when something has electrical potential, it means that it's polarized, that it can yield electricity. That's what it means. It can yield electricity, but it's a negative charge. And I'm going to explain to you in the next few figures why it's negatively charged. So you guys at home, I hope you're watching this lecture on Friday and not the night before the midterm. And if that's the case, please let me know if you yourselves have questions, because I can't answer them live, but I'm happy to answer any of your questions. Just email me or come see me, okay? So the resting membrane potential we know is minus 70 millivolts. How does this occur? Why is it negatively charged relative to the outside of the cell? Well, the first thing that you should know, and if you don't have a background in chemistry, is what is an ion. So an ion is an atom that has either gained or lost an electrical charge. So an ion is a negative ion. It has gained electrical charge. It has gained an electron, right? So an example of an anion is something known as chloride, which we say in chemistry is Cl minus because it's gained that electrical charge. Cl minus. A cation is a positive ion, meaning it has lost an electrical charge. An example of that is potassium, which for those of you that remember your chemical charts is known as K+. Okay, and that's based on the Latin name for potassium, which starts with K. Huh? Kalium. There we go. Kalium. Okay, so chloride is negative, potassium is positive. Why we have the resting membrane potential is because we have an unequal distribution of ions inside and out of the cell. An unequal distribution of these ions. Okay, so bear with me. Here is a cartoon of our happy, friendly cell. I encourage you to reproduce this in your notes. Outside of the cell, the extracellular fluid, we emerged from the sea. So it makes sense that our extracellular fluid is salt water. So what do we know is salt? Salt is sodium chloride. So the ions that make up the majority of our extracellular fluid are sodium, Na+, and chloride, Cl-. Okay, so we have a higher concentration of sodium and chloride ions outside of the cell. Remember that cell membrane? It prevents sodium and chloride from coming in. Inside the cell, we have a higher concentration of negatively charged protein ions. We also have a greater concentration of potassium ions. Okay? So more potassium, more protein ions inside the cell, more sodium and chloride ions outside of the cell. Now, how does this contribute to the resting membrane potential? Oh, this is my little like, haha, let's like have a little cartoon. You guys want to go watch a movie? Okay, nah. A graduate of this class sent this to me, so people do survive and think it's funny. So how do we determine the resting membrane potential? Here are 4 factors, okay? So beautiful exam question. What 4 factors contribute to the resting membrane potential? Boom, boom, boom. Number 1, diffusive forces. Basic science. Ions tend to move down a concentration gradient. What that means is they tend to move from areas of high concentration to low concentration. That's down the concentration gradient. Like a lump of sugar that's put into a glass of water, eventually those individual sugar molecules are going to disperse randomly, and eventually you give your water glass a little shake and you have sugar water. Okay? So ions move down the concentration gradient. In addition, the second factor that determines the resting membrane potential is the differential permeability of that cell membrane. Remember, I told you that it is protective. It typically prevents ions from flowing in and out of the cell. At rest, meaning we've got no stimulation to the cell. It's just sitting in that petri dish. Let's pretend. The cell membrane is totally resistant to the passage of protein ions. Those negatively charged protein ions cannot exit the cell. They're stuck in there. Okay? It is extremely resistant to the passage of sodium ions. So sodium ions could get out of the cell, or get into the cell, but it's very hard to do so. And it is slightly resistant to the passage of potassium and chloride ions. So potassium can get out, chloride can get in, but not a lot. Okay, so you can imagine that the sodium chloride outside of the cell and the potassium and protein inside the cell are pretty much kept there by virtue of the properties of the cell membrane. Okay, so the differential permeability of the cell membrane to these ions is the second factor that contributes to the resting membrane potential. The third factor is something known as electrostatic pressure. Again, I remember learning this in grade 4. Like charges repel, opposite charges attract. Chloride is going to be repelled from the inside of the cell because the inside of the cell is negatively charged, and so is chloride. So they're going to repel. By contrast, sodium is going to be drawn into the cell because the cell is more negative and the sodium is positive. Okay, so that's electrostatic pressure. Again, you guys without the strong science background, don't be scared. This is, remember, grade 4. We learned this with magnets and little iron chips, I remember. Not Celine Dion and Buddy. What's his name? Rene. Of course. Okay, where are we at so far? This is a busy diagram. You don't have it in your notes. I strongly recommend that you take notes as I'm talking. Blah, blah, blah. Okay? So if you don't have a pen in your hand, now is the time to get one in your hand. To summarize where we're at so far. Okay? The inside of the cell has a greater concentration. Anytime you see a bigger box relative to the smaller box, it means a greater concentration. So we have a greater concentration of sodium, negatively charged protein ions inside the cell. It cannot leave the cell because the membrane is completely resistant, right? Or completely impermeable. So it cannot leave the cell, even though electrostatic pressure would be attracting it out. Right? Because the outside of the cell is more positive relative to the inside. Okay? Here's where things get interesting. Potassium is in a greater concentration inside the cell relative to outside the cell. So the concentration gradient is going to push it out. Okay? So the concentration gradient of potassium is pushing it out of the cell. However, because the outside of the cell is positive relative to the inside, electrostatic pressure pushes it back in. So these two forces, diffusion and electrostatic pressure, cancel each other out. Okay? So far we've got much more negative charge inside the cell because of these negatively charged protein ions. And potassium is being kept in because these two charges cancel each other out. Chloride has a greater concentration outside of the cell relative to inside the cell, so force of diffusion is going to push it in. But again, we have electrostatic pressure keeping it out because the inside of the cell is more negative relative to the outside of the cell, so opposite charges, like charges, repel. Okay, so far we're still at zero, meaning, not zero, we're still negative because of those protein ions. Okay, now here's where things get funky. Greater concentration of sodium outside of the cell relative to inside of the cell, so the force of diffusion is going to push sodium in. Oh, what's happening? What's this? Hey, this is curious. Electrostatic pressure is also going to push sodium in because sodium is positive and the inside of the cell is negative. Now remember I told you that the cell membrane at rest is somewhat permeable to sodium. So we're getting a sodium leak into the cell. And if we are all paying attention, this should mean what? The charge changes and it becomes more positive, right? So it's negative. It's kept negative because of those protein ions that are negatively charged. But sodium's leaking in. And it's more positively charged. So hey, how come we still have this resting membrane potential of minus 70 millivolts? Well, the answer is because there is one more factor that contributes to the resting membrane potential, and that is known as the sodium-potassium pump. Remember, I described these pumps. They're a kind of protein that's embedded in the cell membrane, and it involves active transport, meaning it requires energy, or ATP. So what happens is that for every 3 sodium ions that are pumped out, 2 potassium ions are pumped in. So it's trying to get that sodium out of the cell, right? So 3 sodiums out and 2 potassium in. So because of that ratio, there's an imbalance in positive charge, and that maintains the negative charge inside the cell. Okay, so sodium's leaking in, but the sodium-potassium pump is going to pump out the sodium and then replace it with 2 potassium. Questions? I have answers. Yeah?
What supplies the energy?
What supplies the energy? ATP. Yes. It's fine. And don't worry, that's not something that you would need to know for those of you guys that are like, what the heck is ATP? I'm saying this for the benefit of the people who are a little bit ahead of the curve, okay? Just know it involves energy, actively transported. So again, this is a figure that is not from your textbook, so I apologize I can't distribute it. I'm just wanting to go through this one more time because of how critically important it is in understanding the resting membrane potential, so we can understand how that membrane potential is changed. So once again, we have chloride ions that are much more populous outside the cell relative to inside the cell. So a concentration gradient is pushing it in. However, electrostatic pressure is keeping chloride out, okay, because of the like charges repelling. Potassium, same deal, more potassium inside the cell, so it moves down, wants to move down the concentration gradient, but because of electrostatic pressure, it's keeping potassium in. By contrast, sodium is being pushed in both by electrostatic pressure and concentration gradient. And to account for this, we have the nice sodium-potassium pump here that is counteracting all the sodium that's leaking into the cell, and it's pumping out sodium, 3 ions of sodium in replace for 2 ions of potassium. Yes?
Where did you find that picture? Because I actually understand with that, but
Not the other one?
Yeah.
Can you email me, and I will, because sometimes I grab stuff from random sources, and I'll see if possibly I can, if I found it on the web, and then I can just give you how I Googled it. If it's from a textbook, it might get a bit tricky, but I can see what I can do. So email me. I can try and draw it. Worst comes to worst. Okay? Yes. Sorry, what's your name again? Gabriel. Gabriel.
You also mentioned the ion, the protein ions.
Yes.
In all this chain, which is not.
It's not in this figure, no.
Would you mind recapping?
Sure.
what that
So
is?
the big thing with protein ions is because they're not put on here because they're completely impermeable to the cell membrane. So they're stuck in there. So that's really what's maintaining that negative charge, is because we have so many of these negatively charged protein ions and they can't get in or out, that yes, the concentration gradient would be wanting to push them out, and electrostatic pressure would be keeping them in. But because they're completely impermeable, they're not necessarily put on here. But yes, I can, in my ghetto diagram that I could draw for you, I'll include that and I'll share it with you. And you can say, now I understand why Kim went into science and not art. But I'll do it. I'll do it for you guys. Okay. So what all this is, oh, sorry, yes.
No worries. Just like the potassium, like in the sodium-potassium pump, how is the potassium not affecting the overall charge?
So this gets complicated, and in fact, if you go into neuroscience, in third year, you'll learn what's called the Nernst equation, and it will answer your question. But at this point in the game, just know that because the negatively charged protein ions are still more populous, there's more negative charge than the positively charged potassium ions, that it overcomes that, right? So the charge of the cell is always going to be relative to how many negatively charged versus positively charged ions there are in there. So we still have potassium in there, but we just have more negatively charged proteins, so that means it's minus 70.
Even though we're constantly gaining positive ions.
Right. So, like, I can ask you a hypothetical question. If we totally removed all the potassium ions into the cell, what do you think would happen?
If you removed all of them.
All of them. And we just had negatively charged protein ions. What would happen?
Start coming in any,
Or ignore anything else. What would happen to the charge?
it'd be negative.
How would it change? In
Well,
what
it'd
direction?
Like, be more negative.
More negative. There you go. That's all you need to know and understand, is that it would just be more negative. Okay? So the potassium ions keep it from being less negative, if that makes sense. Okay? And what you're going to learn is that it can change, and it does happen that it becomes more negative. Okay? All right, so what all this means is based, oh, sorry.
Do the chlorine ions ever go in?
Do the chloride ions ever go in? There are, yes, there are gates, and we'll learn that they do come in. Yep, it is possible. Scanning the room. Okay, so the neuron is polarized. Okay, that's basically what is all this negative charge, this electrical potential. It means the neuron is polarized. How I remembered this when I was an undergrad is I think the polar regions are cold. The temperatures are going to be in the minus 10 billions or whatever. So polar equals negative charge. Okay? Importantly, how the neurons communicate is by changing their polarity. Neuronal communication, how the signal to change a neuron from the resting state to an active state is done by virtue of the fact that we can change this polarity. We can change the minus 70 millivolts to be minus 50 millivolts or minus 90 millivolts. And that change, just in itself, is a signal to the cell. Okay? That's a way to communicate, like a letter or an email. It's the change in electrical potential. How this is done is by virtue of those inputs into the cell. So remember I said that we took our neuron out and we put it in a petri dish. This is a completely artificial situation. Normally, every cell in the brain has inputs to it. And those inputs, in the form of axon terminals from all those presynaptic neurons, can change the polarity of the cell. So it can change the resting membrane potential. It can occur anywhere in the neuron. More often than not, it's in the dendrites. Remember, the dendrites are those receiving parts of the neuron. But it can occur on the cell body. It can occur on the axon. So here is an example of a postsynaptic cell. Remember, every cell is a postsynaptic and a presynaptic cell. But here, just arbitrarily, this is the cell that's receiving the inputs. And you can see these are the axon terminals from 3 neurons. And you can see that all of these are synapsing directly onto the cell body. Here, you could say it's the dendrite. But more or less, they're synapsing directly onto the postsynaptic cell. And each of these inputs, oh, there's a fourth one. This one is definitely on the dendrite, I would say. Each of these inputs does something to change the polarity of that cell. Okay? So if an input is excitatory, what that means is it changes the cell membrane or the electrical potential to be more positive. Excitatory inputs, more positive. So you can see these are little changes in the voltage. So if we get more positive, that we get an increase in voltage. Increase in voltage. But we can also have inputs that are negative or inhibitory. Okay? So we can have two different kinds of inputs. The postsynaptic potentials are these inputs coming in from the presynaptic cells that occur anywhere on the cell that can change the local membrane potential. So here's the key. They're only changing the cell membrane potential in a very small region of space. The entire cell at rest is anywhere you put your microelectrode, you're going to read minus 70 millivolts. Anywhere on the cell at rest. Okay? But if you have an input from a presynaptic terminal, let's say right here we put our microelectrode right here, and this incoming signal this incoming presynaptic neuron had fired. Don't worry about what that means just quite yet. And we measured the electrical potential here, we would get a more positive reading. But over here in the cell, we'd still get minus 70. Does that make sense? So the incoming postsynaptic potentials only change a local, a very small patch of cell membrane potential. I'll explain this all in greater detail as we move along. So as I mentioned, there are a number of different kinds of synapses that we can have. The inputs, the presynaptic inputs, can synapse onto a number of different regions of the postsynaptic cell. So we can have what's called an axodendritic synapse, where the axon of the presynaptic terminal synapses on the dendrite or dendritic spine. This is the most common, which is illustrated in example A, right here. That's the axodendritic. So here's the axon terminal on the dendrite. We can also have axosomatic. So we're synapsing directly onto the cell body. Or axoaxonic. So this is synapsing onto the axon of yet another input here. And you can imagine this is the mechanism by which we get what's called presynaptic inhibition. So if this cell here is normally acting to increase the cell firing of this cell, but this guy has an inhibitory input, it's going to prevent this cell from communicating to this cell. Okay? Presynaptic inhibition. Yes?
So what's the point?
What's the point?
Because one isn't working then, the other one isn't working.
Okay, let's imagine that this is your I want to eat ice cream cell. This is your move towards the ice cream. This is your diet cell. Block that signal. Go away from the ice cream. Back up, back up. Okay? It's not that simple. I mean, like, we're talking very, like, one cell mediates ice cream. You know, it's not going to happen. You're going to hear specific examples of presynaptic inhibition throughout the term. You just need to know the concept now, and we'll talk about the specifics as we move along.
So it can work like taking, for example, like pills or like coffee or something. So it's the same effect, right?
That's right.
One
So
with
some
inhibit
drugs
and one
work by acting on presynaptic inhibitory mechanisms. Absolutely. Yes?
What's the functional difference between axodendritic and axosomatic?
Excellent question. So for those of you that didn't hear that, what's the functional significance of the axodendritic versus axosomatic? Can I answer that in a few slides, and you'll see. Gabriella.
Could you get an inhibition-like mechanism with the other types?
Yes. So either of these inputs could be inhibitory or excitatory. That's the whole point. And again, we'll hear this throughout the term. What the signal is sending to that postsynaptic cell depends on whether this cell, for example, is in the cerebellum, the frontal cortex. What is the actual signal? Is it dopamine? Is it serotonin? Is this receptor a D1 or a D2 receptor? Is this in the frontal cortex? So this is how the brain manages the complexity of signals, because it's either excite or inhibit. We only got two directions. But what we're going to do at specific times of day or with specific behaviors will depend on where that signal is in the brain, what the neurotransmitter is, and I know I'm getting ahead of myself, and what that neurotransmitter is binding to. So it's really hard to teach this stuff and pick somewhere along the line to start. So I appreciate the questions, and please keep them coming. I just hope that none of you are getting completely confused quite yet. So please do ask questions. Yeah?
So when you're having inhibitory signals, it's not very meaningful just to ask how inhibitory, how much inhibitory.
It is absolutely.
Oh, okay, so it's not digital.
No, the digital is the action potential. The analog is the postsynaptic potential. For those of you that are like, what the hell is he talking about? We'll get there. I saw a hand. Yes?
Would the neural impulse, like integrity, would that change based on the location, like axodendritic versus?
Yes. So you're asking the same question as this young lady. What is the functional significance of the axodendritic versus axosomatic? And we're going to learn for sure what that means, because where the synapse is has a greater impact on the whether or not that neuron becomes active or fires or releases an action potential. Okay? So we will get there. You guys are all like, yes, give me information. And I'm trying to slow you down. Breaks. Okay, the last thing I want to talk about is the dendrodendritic. So we can have dendrites acting on dendrites. And this is quite rare, and what we see is this is found primarily in the olfactory bulb, which is the part of your brain. It's an extension of the central nervous system that sits right above your nose, and it's where you get incoming, smells are coded in your nose, so in the olfactory bulb. And the functional significance of why that occurs in the olfactory bulb, you're not required to know. I'm happy to chat with you about it outside of class. All right, so as I mentioned, we can have two different types of incoming signals, two types of postsynaptic potentials. The first type is the excitatory postsynaptic potential, or an EPSP. Excitatory postsynaptic potential, EPSP. Excitatory postsynaptic potentials lead to depolarizations. Remember, polar is cold. If we depolarize, we make it less cold. So depolarizations make the inside of the cell more positively charged. So we might go from minus 70 millivolts to minus 65 millivolts. We might go from minus 70 to minus 68. Okay? So small changes in the local membrane potential. So we go from minus 70 to more positively charged. Excitatory postsynaptic potentials can occur if that presynaptic input tells, and don't worry about the telling part, we'll get there next week, tells the postsynaptic cell to open one of those specialized gates and allow, let's say, for example, sodium to come into the cell. Because sodium is more positively charged relative to, well, it's a positively charged ion, so it's going to come into that small patch of cell, and it's going to mean that that little space is going to be reading more like minus 65 millivolts. Okay? So here's that rest at rest. If we stimulate, so we make that presynaptic input send the signal to the postsynaptic cell, we open up one of these sodium channels. Sodium comes into the cell, and that local membrane potential will change to be more positive. Okay, so those are depolarizations. That's one kind of signal that can happen. What might be another mechanism to get a depolarizing signal? Not sodium. What are the other ions that can come in and out of the cell? Potassium. Okay, so potassium's inside the cell. So what might be another depolarizing signal? Say that again? Yes. Yes. So we open a potassium channel, and we could allow more potassium in. Okay, what's another one? Chloride. How could we make the inside more negative, more positive with regard to chloride? What would we do? Right, so we have the chloride channel, we open it, chloride comes out. Okay, so you guys understand all that? So people get stuck on the sodium. It can be sodium channels that open, but it could also be potassium or chloride. And the direction of whether they're coming in or out of the cell is what's going to determine whether that's a hyperpolarization, sorry, I got ahead of myself, or a depolarization. So that's an excitatory postsynaptic potential. An inhibitory postsynaptic potential is the opposite. These yield hyperpolarizations, inhibitory inputs. So hyper means above, beyond. So hyperpolarized, increased membrane potential, more negatively charged. And this decreases the probability that that neuron is going to fire. So I think I forgot to say that with depolarizations, you increase the probability that the neuron will fire or yield an action potential. Okay, so these are inhibitory postsynaptic potentials. So here is what you're going to see on an oscilloscope or voltmeter, that the local membrane potential becomes more negatively charged, right? So around minus 73 millivolts. Here are two examples of how we could get an inhibitory postsynaptic potential. We could open up the potassium channel and have an efflux, which means an exit of potassium, making the extracellular side of the membrane more positive. Or we could have chloride coming into the cell, making it, again, more negative inside. So two kinds of signals coming in via the presynaptic inputs. They can be inhibitory or excitatory. These are called postsynaptic potentials. These are not neurons firing. We haven't yet got there. Okay? These are how we can influence whether or not that neuron will fire, is via postsynaptic potentials. I can tell you, I can swear on my life that where all students get confused is the difference between a postsynaptic potential and an action potential. Okay? So they are different. Yes?
So the proteins that allow like potassium and chloride in, the three different kinds, they allow any of the different like the first kind of the protein will let potassium and chloride or sodium in.
So there are channels and gates. Let's imagine we're all thinking about gates right now. Gates can be potassium, they can be chloride, or they can be sodium. They can be any of those. They're different proteins. So if you remember anything about proteins, it's a totally different sequence of amino acids. Right? So we could have a potassium gate, we could have a chloride gate, and we could have a sodium gate. Okay, remember the channels, they let them right in and out. There's no open or closed. Is that what your question is mostly?
Could do each channel, gate, or the
right, so could a potassium, I always call them channels, but could a potassium channel allow in chloride? Is that what you're saying?
I was more asking like if each of them could come in or out through all the same ways.
Efflux or influx? So if we open the gate, could that mean that it could go in or out?
I don't know, now I'm confused.
Okay, think about it. And if you can better rethink the question, then ask me. Okay? Yes?
Can inhibitory messages be selected based on the transmitter?
Yes. Yes. I'm not even going to repeat that because it's way too advanced for all of you just quite yet. Okay?
I'm just wondering what's the medium inside the membrane that prevents the postsynaptic potentials that are local to immediately affect the whole system and such.
Because remember that, so what he's asking is what's preventing the local membrane change from affecting the entire cell? Remember that these postsynaptic potentials occur due to ions moving in and out of the cell. So we're not moving ions in and out of the cell all over the neuron. We're only moving ions in and out of the cell in a small region of space. Okay? So a change in membrane potential directly translates to a change in ion concentration. So if the ion concentration is only changing over here, that means that this place down here is not going to be affected. Make sense?
And that remains over time.
Yes, and what you're going to learn is, in fact, that, yeah, we're going to learn some properties of postsynaptic potentials that will clarify that even further. Gabriella?
I guess another thing that occurred to me is that the opening of the gates is unidirectional.
It can be, right? So, yeah, we're getting more complicated than we need to get right now. Some of the gates are what are called inward rectifying currents, and some are outward rectifying currents. So, again, it depends on the signal, and this is what Rebecca was just asking, is it really is tied to what that presynaptic nerve input is telling that postsynaptic nerve to do. And we'll learn more about that when we talk about neurotransmitters, because it's going to depend on the neurotransmitter and the receptor that it binds to, and that's going to change, that's going to influence what that signal does. Okay? So, I kind of already introduced this idea. Did I put this in your slides? I feel like I took it out. No, it's in there? Yep. Okay, so a little bit about the difference between postsynaptic inhibition and presynaptic inhibition, which is kind of what I already talked about, so I don't know why I didn't take this out. But you can imagine postsynaptic inhibition is essentially this, that we've got, let's say, input A. Presynaptic cell here has an excitatory input, and cell B here has an inhibitory input. So this is essentially B is inhibiting the excitatory effects of A by hyperpolarizing neuron C. Okay? So even though this cell is exciting it, this inhibitory input is going to counteract that excitatory input. The presynaptic input is essentially, again, we've got excitatory synapse A here, and what B is doing is it's directly making contact with the axon of neuron A, and so it's inhibiting the excitatory effects of A by partially depolarizing A. This is not what I'm trying to say. It should be partially hyperpolarizing. Something felt funny about this slide. Apologies. Only the Pope is infallible. I am not. Okay. So it partially hyperpolarizes A. So, yeah, ignore this. I think that's why I got confused. But essentially what that's going to do is mean that this input is going to have a less of an effect, right? Because it's preventing A from depolarizing C. Note to self, change. Sorry, I confused you. Yes?
Are they firing together at the same time?
It could be either that B fires in advance of A, or they can fire concurrently. If A fired in advance of B, it wouldn't make sense.
Oh.
Yes?
So if B is stronger than A.
If B is stronger than A,
inhibitory on C.
that's right. If B is stronger than A, it would definitely inhibit C. Yep. Well, it would prevent A from depolarizing C. What makes one stronger and one weaker? The strength of the stimulus. So imagine very dim light versus very bright light. The amplitude of the signal directly translates, or the input directly translates to the amplitude of the signal. So the stronger the input, the more that presynaptic input is going to fire, and as a result will have a greater influence on the postsynaptic cell. Yes?
Assuming both of them are the same energy, will the end result be the same?
Assuming they're the same energy. What do you mean by same energy?
Yeah, like assuming they both have like the same, like
Strength of signal?
yeah.
In example B or A? Like, so the postsynaptic or presynaptic input?
Like it's for both. Assuming A and B have the same
Strength of signal?
What would the effect on C be the same after those things?
Yes. If we have, in example A, the one on the left, if signal A is stronger, they wouldn't cancel each other out. because we're going to get greater depolarization and therefore a greater impact on whether C will fire. Yes?
When you say strength, do you mean like that it fires more times, or that
So we're not even getting to firing yet. So strength of the postsynaptic, the depolarization, is going to be based on how much ions are we letting in and out of the cell, right? So if it's a really strong input from the presynaptic neuron, and that receptor, it's going to be held open for, and this is very not true, but just for the purposes of explaining this, held open for a longer period of time, so we have greater influx or efflux of ions, so more of that local membrane potential will be, it's going to be even more negative or even more positive.
So it's not more signals?
No, we're not. It's like everybody's getting it is a signal, but let's not get confused again between action potentials and postsynaptic potentials. We're not even on action potentials yet. We're still on postsynaptic potential. So the signal of the presynaptic cell is definitely going to be a release of a neurotransmitter that binds to a receptor and causes those ion channels to open. But the strength of that signal, presynaptic input, is going to change that postsynaptic polarization, hyper-depolarization. These are great questions. I honestly think the more questions you ask, the better, because it means that you're either paying attention or understanding or not shy to ask questions. So I appreciate it. So here are some general properties of those postsynaptic potentials. Okay? And this slide will help make things a lot clearer if they're not clear already. Number one, what we were just saying is the postsynaptic potential is graded. What that means is the amplitude of the postsynaptic potential is proportional to the intensity of the signal. The amplitude of the postsynaptic potential, whether it goes from minus 70 millivolts to minus 68, that's not a very strong signal, right? But if we have a really strong incoming presynaptic signal, don't worry so much about what that means, we can make this amplitude, remember the amplitude is like this, the height of the waveform is proportional to the intensity of the signal. More intense signal, greater amplitude of the postsynaptic potential. Number two, they are decremental. Spare me my art. Okay, here we're getting to some fancy physics terms. Here's my neuron. Do not make fun of me. Oh, here's the axon hillock, and here's the axon, right? Okay, let's say we have an input, a presynaptic input here. Here's that terminal button. And it's located really far from the axon hillock here. And this is what was your name? Amir was asking. So we're changing the local membrane potential right here. Okay, if we have, it doesn't matter if it's excitatory and inhibitory. Only this region, this little patch of membrane is going to be changed because we only have ions that are coming in or out of the cell right here. The postsynaptic potentials are decremental because as it spreads across the membrane, because remember, ions are diffusing, right? They have diffusive properties. They move in a random motion, and they move down a concentration gradient. So they're going to move around here. But as they move, the size decays as a function of the square of the distance. Whew, what the fuck does that mean, right? Everybody's like, ah. Math. What that means is the size of the signal is going to decay as a function of the distance, i.e. the further away it is from the axon hillock, the smaller the change is going to be. So if we have, it's like this, the waveform is declining. Imagine, this is why I've got like 2 tin cans in the background here. I've got a tin can in one hand, and Amir has the tin can in his hand, and I'm talking to him, right? He's going to hear me. But if Amir is in China, he's not going to hear me, right? The size of the waveform is going to decrease as a function of the distance. Okay? That's all that that means. Does everybody understand that? And do not be shy if you don't.
So that means that it's going to be not as strong a signal?
What that translates to is that the further away the input, the less of an impact it has on whether the postsynaptic cell fires. The further away an input is from the axon hillock, which is where all the signals are integrated, the less of an impact it's going to have on whether or not that postsynaptic cell fires. So inputs that are really important, right, on the functioning of this cell, whether or not this cell is going to fire, are going to be right here. These guys are going to have a greater impact on whether that cell fires. Okay? So the nervous system, it's tricky that way, and it's smart. So it's going to wire the system accordingly. These ones are going to have less of an input. These are like my diet cells. This is my ice cream eat cells, right? Okay? Postsynaptic potential, oh, sorry.
Does that actually mean that there are going to be specific like cells, or not cells, but like presynaptic things that are closer to the axon hillock? And because of that, would that have like a specific function?
Yes. Yeah. So what she's asking is, are these inputs going to have a very specific function compared to these? Yes. So this signal is going to be something quite unique, or it could be the same. It could be the same neuron that's branching out its axon collaterals here, right? It could be, or it could not. Okay. They're also rapid. What that means is that those ion channels, they open, they allow ions in and out of the cell, and they rapidly close. Boom. Okay? That's all that that means. It's pretty fast. So essentially, over time, and here you're seeing voltage change. Here, like we might get this. Okay? It's going to rapidly go back to the resting membrane potential. So ultimately, we say that postsynaptic potentials have passive cable properties. Does anybody want to take a stab at what it means to have passive cable properties? Yes?
They're not active until something makes them active?
In a manner of speaking, I'll give you that one. Yes. Can I have somebody clarify more on what it means to have cable properties?
To kind of mean like you can pass along the signal without having to do it.
Yes, that's what the passive part means. What does the cable part mean? I'll give you a hint. I already gave you an example. Think about cables, right? Okay. Fiber optic cables or whatever cables. Yep.
One fires have multiple membranes, and the fire fires more and more.
No. Anyone else? Yes.
When you change the concentration, it basically sets off a chain reaction. So it continues down.
Again, in a manner of speaking, I'm thinking even more simple than that. No. So we're talking about postsynaptic potentials.
The signal has less of an impact because it goes farther down the axon, so it's like decremental.
Okay, not farther down the axon, but further along. Yeah, so it relates to mostly this property, these properties, right? This is what it means to have cable properties. That it's a graded and decremental phenomenon because the signal decays as a function of distance, like a cable. Okay, the further away the cable is, the less of an impact it has. Okay, that's what it means to have cable properties. And what you'll learn with action potentials is they don't have cable properties. These are also known as analog. So another way of looking at this is an analog. It's a direct representation, a one-to-one representation of the signal. All right, I'm going to take you through this interim summary. So where did we learn today? The inside of the cell is more negative than the outside of the cell. And this is what's called the resting membrane potential, which is at minus 70 millivolts. We learned that this is due to the imbalance of ions inside versus outside the cell. And as a result of that, also the 4 factors that contribute to this imbalance. So we maintain the imbalance due to, what are the 4 factors? What? Force of diffusion, number 1. Electrostatic pressure, number 2. Sodium potassium pump, number 3. Last one. We already got that one. Differential permeability of the membranes. Excellent. This means that the neuron is polarized. It has electrical potential. It can change. And how it changes is via the postsynaptic potentials, which are local, graded, rapid, and decremental changes in the membrane potential. They can be more negative, hyperpolarizations, or they can be more positive, depolarizations. Depolarizations increase the probability of the neuron firing, i.e. releasing an action potential. Hyperpolarizations are inhibitory signals. They decrease the likelihood that that cell is going to fire. What we are going to learn in possibly the next 5 minutes, no, I'm just kidding. I might introduce it, but what we're going to learn mostly next class is the fact that if the sum of all the depolarizations means that the inside of the cell reaches a membrane, a voltage of minus 50 or so millivolts, which is the threshold of excitation, this is going to trigger an action potential. So if the sum of all the incoming excitatory inputs means that the inside of the cell becomes minus 50 millivolts, which is the threshold of excitation, it's like the trigger point, then this is what's going to cause that neuron to fire or release an action potential. And that is known as the threshold of excitation. I think what I'm going to do is, rather than start you with the action potential, which is crazy, is I'm going to take the next 5 minutes to answer questions. I'm also going to remind you that this Friday, what we had scheduled was a review as well as a little bit of a biological sciences primer. Because of the power outage, what I've done is I've made, I'm going to keep teaching, so I'm going to teach the rest of neurophysiology next class. And instead, next Friday is going to be a biological primer, a review for the midterm, and an open Q&A. So 2 of your TAs are going to do that, Rob and Catherine. Anna.
Next Friday's
CU day? Never be mind. I meant the following Friday. Don't come here on CU day. You're meant to be studying. So, anybody have any lingering questions? Yes.
Why is polarization
inhibitory? Because in order for a cell to fire, the inside of the cell has to reach minus 50, which is more positive compared to minus 70. So depolarizations make the cell more positive. Hyperpolarizations make them more negative, right? So we're going in the wrong direction. Right. Yes.